scientists are working for the development of new drugs for the new dosage form in order to improve the viability and improve the effect of the drug. The process of drug discovery involves the identification, candidate synthesis, characterization, screening, assay for therapeutic efficacy. Basically, this is a time-consuming process, and there is large amount of investment is required for to bring a product in the market. This is the flow chart of drug discovery and development. First of all, there is identification of disease, and there is the protein isolation. It takes approx 2 to 5 years. After that, there is a need to find a new drug effective against disease protein. It takes 2 to 5 years. And after that, there are pre-chemical testing that are performed on the animals. Formulation is developed, further scale-up is there, and human clinical trials take place up to 2 to 10 years. FDA approval, 2 to 3 years. So, in brief, approx 15 years are required, and a large amount of investment is required to bring a drug that should be available in the market. These are the phases of drug discovery process. First one is target identification, target validation, compound screening, drug optimization, preclinical trials, and clinical trials. And after that, there will be IND will find if that will be approved for the government and the concerning government that will be available in the market. There is a brief summary of the target selection of the inventory process in this research process, target selection, cellular and genetic targets, genomics, proteomics, bioinformatics, and after that, there is several steps involved. Synthesis and isolation, combinatorial chemistry, assay development, high throughput screening, medicinal chemistry, includes library development, SPRC studies, in silico screening, chemical synthesis. In vitro studies includes drug affinity and selectivity, cell disease models, MAO, lead candidates refinement. And after that, there are preclinical studies. It includes animal models of disease state, behavioral studies, functional imaging, ex vivo studies. And after that, there are the clinical trials that are performed over the human beings. And this is the brief pipeline of the drug discovery process. Target identification, validation, lead identification, lead optimization, preclinical development. These are the phase first, phase second, phase third, phase four clinical trials. And the approx investment process is approx $467 million. And there are 10,000 compounds that are initially selected in the preclinical trials. There are only 250 compounds in the place. And for the clinical trial compound, and FDA approved only one drug that will be to be marketed, to be launched in the market. 6.5 years time taken for the preclinical studies, 7 years for the clinical trials, and 1.5 for the approve and review process. So I welcome once again all the participants, all the speakers of the different countries, and I expect that this webinar will be fruitful for all. Our participants will get knowledge that will be enhancing of the knowledge. So I would like to invite Ms. Basta Negi to continue this event. Thank you.
Good morning, distinct guests, respected dignitaries, esteemed speakers, and dear participants from across the globe. I am Vasudha Negi, Assistant Professor at Samrat Prithviraj Chauhan College of Pharmacy. Today, I feel truly delighted and deeply honored to serve as the anchor of this esteemed international conference. The theme of today's conference is Revolutionizing Pharmacy, Addressing Drug Development, Safety Concerns, and Global Health Challenges. This theme reflects the urgent need of our time, where pharmaceutical science is not only advancing rapidly, but also taking responsibility for ensuring safer drug development, improving patient outcomes, and addressing critical global health issues. We are gathered here today with a shared vision to exchange knowledge, explore innovations, and contribute meaningfully to the future of healthcare and pharmacy. This is a multidisciplinary conference dedicated to transforming the field of pharmacy by advancing innovative drug discovery and development, strengthening medication safety and pharmacovigilance, and addressing pressing global health challenges through research, technology, policy, and international collaboration. It emphasizes improving how medicines are created, tested, regulated, and delivered while ensuring they are safe, accessible, and effective for populations worldwide. Once again, I warmly welcome you all to this enriching academic platform and look forward to a highly insightful and inspiring day ahead. Ladies and gentlemen, get ready for an exciting and insightful session as we move forward with our first distinguished speaker. It's my absolute pleasure to introduce Professor Dr. Mahmood Arba from the Faculty of Pharmacy, Holu University, Indonesia, a passionate researcher and an academic inspiring academician. Professor Arba has made significant contributions in the field of pharmaceutical sciences. His work, especially in advancing modern approaches to drug discovery, reflects innovation, dedication, and a deeply commitment to scientific excellence. His expertise in computational studies has opened new doors in making drug development faster, smarter, and more efficient. We are truly delighted and honored to have you with us today, Professor. A very warm welcome to you. So, in simple terms, computational studies are transforming the way we discovered medicine. Instead of relying only on traditional methods that can take years, scientists now use advanced computer based techniques to stimulate, analyze, and predict how potential drugs will behave. Today, he will be speaking on a topic that is shaping the future of healthcare and research: the role of computational study in accelerating drug discovery process. This not only speeds up the entire process, but also improves accuracy, reduces cost, and bring life-saving medicine closer to reality much faster than even before. Sound fascinating, isn't it? So, without any further delay, let us dive into this innovative world of science. Please join me in extending a big round of applause as I invite Dr. Mahmood Arba to take the stage. The stage is all yours, Professor. Professor Dr. Mahmood
हाँ हलवाई नहीं क्या ऑनलाइन नहीं है क्या मैं तो अनाउंस करूँ कैसे
Professor Dr. Mahmood Arba, please, sir.
हेलो Up next, now,
there is some technical issue. So I am announcing the next distinguished speaker, Professor Ahmad Nazib from the Faculty of Pharmacy, Universitas Muslim Indonesia. Professor Nazim is a renowned academician and researcher widely respected for his contribution to pharmaceutical sciences, especially in the area of natural products and traditional medicine. His work beautifully bridges the gap between age old knowledge and modern scientific innovation. We are truly honored to have you with us today, sir. A very warm welcome to you. Today, he will be speaking on a truly captivating topic: ethnopharmacology meets innovation, exploring anti diabetic mechanisms of Sulawesi botanicals. This topic brings together the wisdom of traditional herbal medicine and the power of modern scientific research. Ethnopharmacology allows us to explore how indigenous plants, especially from biodiverse regions like Sulawesi, have been used for generations to treat diseases such as diabetes. By combining the traditional knowledge with innovative scientific approaches, researchers are uncovering the mechanism behind these natural remedies, making them more reliable, effective, and globally relevant. It's a perfect example of how tradition and innovation can work hand in hand to address one of the most pressing health challenges of our time. Sounds truly exciting and insightful. So, without any further delay, please join me in giving a warm and enthusiastic round of applause as I invite Professor Ahmed Nazib to take the stage. The stage is all yours, sir.
Okay, thank you very much. Is my sound clear, Dr. Negi? Okay, is that clear? Is my sound is clear?
Yes, sir, it's clear.
Okay, thank you very much, Dr. Negi, and especially from Dr. Kumar to invite me again on this international webinar. This kind of honor to join with you, all of you in this room, even online process. So I will share about my research. It's clear?
Yes, sir, it's clear.
You can see? Okay. I will share about ethnopharmacology myths, innovation, exploring antibiotic mechanism of Sulawesi botanical. And once again, from team international seminar from Samrat Prithviraj Chauhan College of Pharmacy. This time, I will introduce my presentation that Indonesia is very large archipelago. It has 5 big islands. One of 5 is Sulawesi. Now is Indonesia have a second mega biodiversity country. It's cover 15.5% of the world flora. Then Indonesia have kind of ancient traditional remedy as jamu, like in India as Ayurvedi and Japan kampo. This is a Sulawesi Island, the 4th largest island in Indonesia. The locate is so special because it located on Wallace line between Australia zone and ASEAN zone. The Wallace lines divided into 2 regions, region Australia and region Asian. This is higher number of endemic plants, especially in Sulawesi, but the poorly study are in South Asia. So it have a high possibility to discover novel unique bioactive compounds. So it will need totally a phytochemical study. From that background, there are many medicinal plants. We take 4 from Goa region: Antidesma buneus, Syzygium tumini, and Syzygium malacens. It's collected in July 2024. And the one is Cordia mixa from Enrekang, the same time, July 2022-24. We know that diabetes still has a prevalent high in the world. There's a rising global burden, and we know this from International Diabetes Committee in the Southeast Asia, especially on generally in Asia, is there are patient of diabetes mellitus. Current therapy is effective, but costly and has adverse effect. So we can get the drugs or therapy for safe, affordable, and more innovative from the research innovation. If we compare from the drugs and herbal therapy, there are synthetic chemical, high cost, and has a side effect. Beside the herbal remedies, they are from natural compounds, affordable, and less side effect. So ethnopharmacology can bridging from the conventional drug and the herbal remedy. So traditional medicine can get can give data become modern science as a therapy in diabetes mellitus. So research object on our research is identify bioactive compounds, evaluate alpha glucosidase inhibition, compare with acarbosa as a chemical therapy used in alpha glucosidase inhibition, and explore drug development potential from the plant and with mechanism of enzyme inhibition, and compare it between a natural product and a chemical or conventional drug. We extract the sample from the crude drug with ethanol 95% with maceration methods, and it gives yields highly from Syzygium malacens 2.97%. After that, we screening the phytochemical profiles, and we found from the samples is hello. We find samples from flavonoids as antioxidant on the samples, and terpenoids, it potentiates hypoglycemic, then saponin to secretion of insulin, and the tannin to enzyme inhibition. So natural product often provide multi-target drug activity. After biopsy results and alpha glucosidase on this sample with microarray system, and we found that potent alpha glucosidase inhibition with acarbosa as positive control, the IC50 value is 6.85 ppm. Meanwhile, in extract IC50 highest from Antidesma buneus with concentration 5.66 ppm. Then the other extract lower than 6 ppm. So if we compare the IC50 from the activity to inhibit alpha glucosidase is the 4 samples is potential or more potential than acarbose is a positive control. So all extract outperform standard as drug activity. The mechanism and safety, we can know that from linear work, kinetic enzyme, so that the four sample has inhibition is non-competitive. It means can inhibit enzyme on the different side of the substrates. From the discussion for in silico, the binding site it's different from binding set of the native ligand. So it decrease carbohydrate digestion. So from the postprandial glucose, decrease the glucose concentration in the blood glucose and is decrease from oxidative stress. So from the addressing global challenge, we can have a change to drug development for faster discovery, and the safety will be fewer side effects, and the global health, it will be affordable access, and the innovation for integration of tradition mechanism of traditional medication with conventional therapy in diabetes. So ethnopharmacology can integrate botanical and chemistry and pharmacology in drug discovery. From this addressing global challenge, we have available research for collaboration. So hopefully it will be international cooperation between institutions from Indonesia or from India or from Turkey and so on. So as a conclusion, we know that Sulawesi plants for the four especially has a strong anti-diabetic candidates. The anti-desmobenus is most potent to support safe and accessibility therapeutics and to contribute to global health equity. So I will give big thanks to the Catalyst Research Grant Schemes under the RTPM Dicty 2024 is our part of our time, our team to conduct this research, and hopefully you can discuss from this short discussion, hopefully, and thank you very much.
Thank you very much, sir, for that wonderful and thought-provoking presentation. Let's give a big round of applause for our esteemed speaker. Now, get ready for an exciting and insightful session as we move forward with our next distinguished speaker. It's my absolute pleasure to introduce Professor Dr. Mahmood Arba from the Faculty of Pharmacy, Holu Olio University, Indonesia. A passionate researcher and an inspiring academician, Professor Arba has made significant contribution in the field of pharmaceutical sciences. His work, especially in advancing modern approaches to drug discovery, reflects innovation, dedication, and a deep commitment to scientific acceleration. His expertise in computational study has opened new doors in making drug development faster, smarter, and more efficient. We are truly delighted and honored to have you with us today, sir. A very warm welcome to you. Today, he will be speaking on a topic that is shaping the future of healthcare and research, the role of computational study in accelerating drug discovery process. In simple terms, computational studies are transforming the way we discover medicine. Instead of relying only on traditional methods that can take years. Scientists now use advanced computer-based techniques to stimulate, analyze, and predict how potential drugs will behave. This not only speed up the entire process, but also improves accuracy, reduces cost, and brings life-saving medicines closer to reality much faster than ever before. Sound fascinating, isn't it? So, without any further delay, let us dive into this innovative world of science. Please join me in extending a big round of applause as I invite Professor Arba to take the stage. The stage is all yours, Professor.
Okay, thank you, moderator. Can you hear me?
Yes, sir.
Okay. So, thank you to the committee who has invited me to this wonderful webinar. Please let me show my PPT. Uh, Sorry, I don't find.
He is open.
Sir, please open your camera.
Yeah. Sorry, how to show the PPT?
Sir,
Hello?
there would be an option on the upper hand right side.
Button.
So with the option of share. Sir, after clicking the option on share, then you have to click on the share screen.
What share? Okay. And then
So then click on screen.
um,
Sorry, there is some technical issue. Now, I am introducing the next distinguished speaker, Professor Dr. Husni Kalyar from the Faculty of Pharmacy, Department of Pharmacognosy at Edge University, Turkey. It's my great honor that Professor Kalyar is a highly accomplished academician and researcher with extensive expertise in pharmacognosy and traditional herbal medicine. Her work has significantly contributed to understanding the therapeutic potential of natural products while also addressing important aspects such as safety, efficacy, and regulation. Her dedication to advancing knowledge in this field and bridging the gap between traditional practices and modern scientific standards is truly commendable. She is widely respected for her research, academic excellence, and valuable contributions to the global pharmaceutical community. We are truly privileged to have such an eminent personality with us today. Ma'am, a warm welcome to you. We are delighted to have you here with us. Today, she will be speaking on a very important and relevant topic, traditional herbal medicinal products in Turkey, regulation and interactions. As the use of herbal medicines continues to grow worldwide under the influence of globalization, innovation, and technology, Turkey remains one of the most hello. Hello. Hay Allah. Is there a connection problem?
Sorry, Professor Khayalar, I hear you. I hear you.
Oh,
You can present.
you-
You can present your
You hear?
presentation.
Okay, okay. Thank you very
I
much.
hear you.
Thanks from Izmir, Turkey.
Okay, okay.
I hear you. I hear you. I think there's connection problems. Hello, everyone. Shall I start with my presentation? Do you want me to share my presentation?
Okay, Professor. I
Okay,
hear you.
okay, okay. I
You
was
can,
waiting.
sir. You can, sir.
Okay, okay. I'll share my presentation and I'll start with. Okay, okay. You see my slides?
Okay, that's crystal view.
Okay, okay. So
Continue,
let me
Professor.
start. Okay, okay.
Better view
Hello
of
to everyone
presentation to you.
from Izmir, Turkey. I would like to begin my remarks by thanking to Professor Kumar for inviting me to this valuable and informative webinar. Today, I'll provide brief information regarding the legislation on traditional herbal medicinal products in Turkey, especially those are ones with certain licensed traditional herbal products and their potential herb-drug interactions, and some examples with dietary supplements interactions. At this point, I'm
presenting here the logos for Turkish Medicines and Medical Devices Agency, which operates under the Ministry of Health in Turkey.
Thank you,
And
Dian.
you hear me?
I hear you, Professor.
Okay,
Continue,
okay.
continue.
At this point, here you see the logo for licensed traditional herbal medicine products. Let me briefly define what is traditional herbal medicine. Traditional
Sudah.
herbal medicine is the sum
Anu,
of
speaker.
total of
Sudah. Tapi masih ini masih.
knowledge, skills, and practices
Speaker
based
karena
on.
di Turki masih pagi sekali tuh.
I hear you. Sorry, I hear some others' voices. Please let me go on with the presentation. Okay. Thanks for your attention. It's the sum of total knowledge, skills, and practices based on theories and experiences indigenous to different cultures. And they are used to maintain health as well as prevent, diagnose, improve, or treat physical and mental illnesses. Sometimes in other countries, they use the term complementary medicine or alternative medicine interchangeably with traditional medicine. But as a result, it is the sum of different cultures for using the prevention, diagnosis, improvement, or treatment of physical and mental illnesses, the herbs. When we say herbal medicine, the issue is the standardization of certain herbal drugs. So we have to confirm their identity, quality, and their pureness. So when standardization is mentioned, what we look for the first time is the pharmacopoeia monographs. As we all know, pharmacopoeia monographs serve as national laws, while every country has its own pharmacopoeia. As we know, Turkey officially published Turkish pharmacopoeia in 2017, and it's adapted from European pharmacopoeia. And we use this pharmacopoeia at the moment. To answer what constitutes a traditional herbal medicine product is the drugs and preparations that have been in use for at least 15 years within European Union countries and of course also Turkey, and for at least 30 years globally for other continent countries. For the licensing of these traditional products, there is no requirement for new clinical trials. That's the point, as they rely on the transfer of knowledge and experience accumulated over many years. This is the European legislation for the licensing of traditional herbal products, and they must be complied with good manufacturing practices. All the herbs must comply with GMP rules, and they must all be submitted by physiochemical, biological, and microbiological tests. But as all we know, there are potential risks in herbal products. So, for example, the inability to characterize all compounds, or also plants used in preparation of these products, the collection time of the plants, the seasonal changes, the geographical location, or part of the plant's use are important parameters that affect these quality standards, as they are all complex mixtures. So we have to standardize, and potency and safety is also important at this issue. And the other risks are social life risks. For example, the advertising of such products via media and internet, selling them with false and misleading, may cause misuse, and recommendation of these products between others or without consulting a pharmacist or doctor is also a serious public health problem when using herbal products. As we know, undesirable effects may also occur due to wrong selection or adulteration with other herbal drugs or compounds, which are frequently encountered, and also they have serious consequences. So we need actually we need a globally harmonized regulation, not by country, but we need globally harmonized regulation for these such products. In our country, in Turkey, the Ministry of Health training programs for physicians on phytotherapy and homeopathy, and also complementary medicines such as homeopathy, for example, or acupuncture, have started, and it's anticipated that the risk in question will be minimized in short time. In Turkey, in my country, herbal products are licensed under two authorities. One is Ministry of Health, and the other one is Ministry of Agriculture and Forestry. Ministry of Health give license to herbal medicines and also traditional herbal medicinal products, and Ministry of Agriculture and Forestry give license to food supplements. When we compare these supplements and traditional herbal medicines, food supplements cannot have indications on their boxes, but traditional herbal medicines are used to intend to diagnose or to prevent some illnesses or so this is the law which was in the legislation in 2010. It was published in our official Turkish Gazette, and it is renowned again in 2023. It's adapted, and the last version is published in 2023. And the purpose of this regulation is to ensure that the traditional herbal medicinal products have been licensed and they have the desired efficacy, safety, and required quality. It's the point here. So this regulation have some modules. And also, in addition, in 2023, aromatherapeuticals and vitamins, minerals added in medicinal products are also evaluated within the scope of this regulation. But there are some excluded. For example, medicinal preparations are excluded. Medicinal products intended to be used in research and development studies are also, and semi-finished products are also excluded from this legislation. Plants marketed as food also, including food supplements, also excluded. So there is confusing in here. There is confusing issue in here, because when we say food supplements, the only point here is that they do not indicate indication in their boxes. So food supplements, when we define them, we determine daily intake dose. They have also concentrates or extracts of some bioactive substances similar to traditional herbal medicinal products. So one authority might be enough, and this must be the Ministry of Health, in my opinion. So no indication can be specified in the label of food supplements, but in traditional herbal medicinal products, we use the label that they have indications. In our country, herbal products, as I said before, are regulated like in European Union regulations. And there are 5 modules. It's aimed to compile the proof and efficacy and quality and safety of the finished herbal medicinal product. For example, in module 3, there are results of the active ingredient and finished products analysis. And we all use pharmacopoeia standards, and the monographs. If the analytical methods are not included in pharmacopoeia, they should be validated methods for the dossier. And for example, module 4 covers non-clinical data of the finished product because according to definition, it's not obliged to give clinical data. Only the use of at least 15 years in European Union and 30 years of usage other than the other countries is enough. So in case of any doubt regarding the effectiveness, they must give preclinical and clinical data if needed. So here you see some products which are traditionally herbal licensed, and you see here the logos on the boxes. And these products are designed and intended to be used without physician supervision, prescription for diagnosis or treatment monitor. You can go and buy from pharmacies if you wish. And there are preparations that are used orally, externally, and by inhalation here. And they are only sold in pharmacies, and promotion and advertising through media and to other are prohibited. So they are just like medicines. They are regulated just like medicines. And here you see again the logo, which is for traditional herbal medicine. It is designed for the awareness of patients and especially pharmacists. Here you see the website for Ministry of Health about the licensing of traditional herbal medicine in Turkey. And we have some important lists about these. There is a list that are licensed medicinal plants, and you here see all the information about drugs, about the ingredients, and also about the short report of these products. And here you see some licensed products in Turkey. And for example, one includes Pelargonium pseudoides radix. The other one includes Centauri herba radix, and the other one with Hedera helix, and the other one Gentiana radix, Primula flos. Here are the list of our traditional herbal medicinal products. As you see, there are different constituents in all products. And as herbal, we always have in mind that they are harmless. As they are herbal, you can use in any way you want. This is really wrong idea, because they have really harm effects if you use in wrong conditions. So here, for example, again, Hedera helix or Centaria herba here, Passiflora, and Silimarin, we have Silibum marianum preparations, and other, for example, Equiseta herba, Ahillia millefolium here. There are, for example, Punica granatum, or some oil preparations, for example, Cucurbita semen oil, Linum ustatissimum, linseed oil, or Oleum vitis vinifera. Or ginkgo, for example, this is also licensed as traditional herbal medicinal product here. And I also want to give some examples of herb-drug interactions for some of these examples. For example, here we see cerebil that includes ginkgo biloba dried leaf extract, or Punica granata, or Nigella sativa seed oil. For example, Aceta racemosa, Smifuga racemosa rhizomes, and Linum ustatissimum, linseed, Cucurbita semen, Silibum marianum semen. Here you see, and the other ones, Passiflora, Hedera helix. These are the preparations that are licensed under traditional herbal medicinal products. And of course, the herb-drug interactions are really serious problem. So the factors are patient related factors or quality of plant material and plant product, or pharmacological properties or pharmacokinetic profiles, but the interactions, some interactions are really harmful for the user. For example, ginkgo biloba dried leaf extract is used against Alzheimer type demence, vascular demence, or mixed forms of demence syndromes. It's also used against vertigo, tinnitus, but it is contradicted against hypersensitivity. Risk of intracranial bleeding is occurs. Also, clotting diseases problems might occur, and it is ginkgo biloba preparations are contraindicated against children, diabetes patients, against hypertension patients, against who have coagulopathy or retinal bleeding or retroperitoneal bleeding. It's all contraindicated to these patients. The ginkgo biloba have the risk of increasing bleeding with anticoagulants, antiplatelet agents, and low molecular weight heparins. So we can also give some examples. For example, with anticonvulsants, it increases the epileptic seizure. For example, with serotonin reuptake inhibitors, it increases also serotonin syndrome. With insulin, it also have the risk of hypoglycemia and hyperglycemia. With thiazic diuretics, it is also effective on blood pressure. With aminoglycosides, it has ototoxicity risk. So we can give more examples. For example, with cyclosporin or midazolam, with benzodiazepine groups drugs, it is really dangerous to use ginkgo preparations. So in acetaminophen, for example, in anticonvulsants, in aspirin, warfarin, valerian, in cyclosporin, in antiepileptic, theophylline, doxorubicin, cisplatin, in concurrent use with ginkgo biloba, it could be really harmful. And for example, the other example with climadinon, which includes Actea racemosa rhizome, it's used again in menopausal complaints, as we know, and there is brief product information in the box of this product. And patients who have breast cancer or who have hormone-related tumors must consult to a physician before using this product. As we say, there is no obligation for the physician device, but they must ask for physician if they can use these products or not. And here you see the contraindicated problems. Here you see, and I use Eric's Media Pharma for these knowledge of interaction problems. For example, Smith-Wiggle racemosa might gastroenterite, nose, vomiting might cause these, and also liver problems. And when you use Smith-Wiggle racemosa preparation at high doses, it may cause hypotension, headache, vertigo, or blurred vision. And it has also, of course, interactions with other all these substances. And the other one is Passiflora incarnata. Here you see one example of this traditional herbal medicinal product. And Passiflora is also not regarded to use in children under 12 years of age. For example, it also have interactions with synthetic sedatives. You must have physician advice to use Passiflora. So, and you may have adverse effects with Passiflora, such as nausea, ventricular tachycardia, or drowsiness. And it also have, of course, sedative effects, and it may potentiate the sedation of these sedatives. And also coumarin, warfarin also is have interactions with Passiflora incarnata. And you here see the Eric's Media website for Passiflora or the other ones. Here you see Alora Ultra tablet at Eric's Media website. It's interactive site. And the other one, Umcavino, Pelargonium pseudoides dry root extract. It's used to relieve symptoms of acute and upper respiratory tract diseases such as cold, bronchitis, or sinusitis, or runny nose, or congestion. We use Pelargonium pseudoides extracts. And here you see the Eric's Media website for Pelum oral solution. But it also have adverse effects and interactions. For example, possible adverse effects might be stomach discomfort, or it might interact with anticoagulant drugs. Before surgery or dentistry, its use should be discontinued before 2 weeks, for example. And liver damage might occur with long-term use of this Pelargonium pseudoides products. The use of Pelargonium pseudoides should be under supervision of the that have liver disease problems or the ones with excessive alcohol consumptions, or also the drugs that are metabolized in the liver. They must be careful with using this Pelargonium pseudoides preparations. And here you see the other preparation. And we have heterohelix here, heterohelix preparations, liquid extract, for example. It's also used again, yani upper tract infections, respiratory tract infections. But it should not be used at the same time with opioid antitussive preparations. Here you see again the Eric's Media site for this heterohelix preparations. And the other one, Harpogophytum procumbens. It's also used again against back pain, or it's used for symptomatic treatment of painful osteoarthritis. So here also it has interactions with other preparations. For example, medications for all types of infections, anxiety or depression preparations, medications, for example, cancer medications, erectile dysfunction medications, or migraine medications. Interactions might occur with Harpogophytum procumbens. And the other ones, I want to only mention one or two examples. For example, red pepper, capsaicin, it has capsaicin in it, and it's used against muscle and joint pain in neck, shoulder. It's externally here used, but we may have preparations which are used internally. So in orally preparations, it might have interactions with barbiturates, theophylline, or monoamine oxidase inhibitors. Hypertensive attack might occur with red pepper oral preparations, and also it's harmful to use with anticoagulants. And the other one, Linum ustatisum, as we all know it as linseed oil, and also here it might also delay the absorption of some drugs or medicines. It may also cause diarrhea. So in another example, Sinameki, Senna angustifolia, used against constipation, but also it has interactions with digoxin, thiazide, cardiac toxicity, licorice, corticosteroids. If you use with cardiac supplements, you may have hypokalemi with Sinameki preparations. And with long-term use, you may have gastrointestinal complaints, also electrolyte disturbance, also albuminuria, hematuria, and also pigmentation in the intestinal mucosa, and just the same as Xinameki or Cassia angustifolia, the same as aloe, also with digoxin, thiazide group diuretics, cardiac toxicity might occur, electrolyte imbalance and hypokalemic might occur. It should not be used for more than 2 weeks. And the other one is again Glycyrrhiza glabra, licorice, or in English, especially the patients with using antihypertensive drugs might be harmful when they use licorice preparations. And the other one, Vitex agnus castus, and it's contraindicated in estrogen and progesterone tumors, and it has side effects and interactions, especially with dopamine antagonists and oral contraceptives. And we all know garlic, which lowers blood pressure and which is protective against cardiovascular disorders. And it should be discontinued before 10 days, for example, before surgery. And it has also interactions with warfarin, aspirin, or hypoglycemic drugs. And echinacea used for immune stimulant, and it should not be used more than for 8 weeks. And it has interactions with cyclosporine, methotrexate, ketoconazole, caffeine, warfarin. In 2 weeks, in acute infections might occur because of Asteraceae allergy. And the other one is Zingiber officinale. It is used in the prevention of some such as diseases, dizziness, nausea, and vomiting associated with motion sickness. It is also contraindicated with using hypoglycemic drugs, antihypertensive drugs, cardiac glycosides, and warfarin, for example. It should be also discontinued at least 1 week before surgery. And the other one, Silybum marianum, for liver and gallbladder problems, and for lack of appetite, for dyspepsia, this Cinnaris colimus also used. And both Cinnaris colimus and Silybum marianum are from Asteraceae family, so we may have Asteraceae allergy. Also, the ones with bile duct and intestinal obstructions are also contraindicated. And interactions with glycoprotein P and CYP enzymes might decrease or increase the absorption of such compounds. And the other one, Seroneura repens, for example, it's used for difficulty in urinating, and it has also some contraindicated problems. For example, finasteride with androgenic agents, salpamento preparations are contraindicated also with anticoagulants. And the other one, fucus vesiculosis, it is contraindicated when you use hypoglycemic drugs or central stimulants. It should not be used for long term, as for example with levothyroxine, hyperthyroidism might occur. As a conclusion, we all have responsibilities to eliminate the false belief that herbal products do not pose safety risks because they are natural. So we have responsibilities to ensure that effective, safe, and quality products offered to the market. And we have to use the right product in the right amount, in the right time, and for the right person. Also, drug authorities should update their regulations and while contribute to this goal by conducting marketing surveillance and control of products. Patients should consult healthcare professionals, especially to pharmacies, and international harmonized and accepted regulation must be regulated, and it will contribute to the safe and safety of these products. And regarding the use of these herbal products, it's not correct to believe that they are harmless or they are natural. And it should not be forgotten that the only professional group, especially in Turkey, have the training on herbal medicines and phytotherapeutics are the pharmacists. The pharmacist is the first knowledge for advisor on natural products. And here you see some sources, especially books or websites, for example, EMA monographs for final assessment reports of some herbal drugs. And you here see ESCOP monographs, PDR, WHO monographs. In Turkey, we have FFD monographs and World Health Organization monographs. These are the sources as books we can consult. And the other ones, for example, Edix Media is really an informative, interactive knowledge source. And also, I want to thank to Professor Bijan Kılçak. She is at our department, and she is the member of the Traditional Herbal Products Commission. And Professor Levent Üstünes is the founder and the editor-in-chief of Edix Media. He got retired from our faculty. And also, I want to mention some leading names of scientific pharmacy about pharmacognosy, especially Sarım Çelebioğlu, Professor Asuman and Turan Baytop, and Professor Nevin and Mekin Anker, for example. They are doyens of this pharmacognosy and phytotherapy. Also, Ali and Professor Liz Meriçli, and Professor Necmettin Zeybek, and also his son, Professor Ulvi Zeybek. Also, I want to thank to all you for your kind attention. And this is my colleagues and our laboratory, you see, and my daughter. We all thank you for your attention.
Thank you very much, ma'am, for sharing your such valuable insights with us. I'm sure everyone has learned a great deal with your presentation. Ladies and gentlemen, what an incredible journey of knowledge we are expressing today. Each session is bringing fresh insights and expanding our understanding in truly meaningful ways. And now it's time to move ahead to another exciting and forward thinking session that blends nature with cutting edge technology. It's my great pleasure to introduce our next esteemed speaker, Dr. Abul Kalam Azad from the Faculty of Pharmacy, MAIWP International University College, Batu Muda, Kuala Lumpur, Malaysia. Dr. Azad is a distinguished researcher and academician known for his innovative contributions to the field of pharmaceutical science. His work particularly stands out in the area of advanced drug delivery systems and nanotechnology, with a strong focus on integrating natural system into modern system. He has been actively contributing to the development of smarter and more efficient therapeutic solutions. His dedication to research, innovation, and academic excellence make him a highly respected figure in this field. We are truly honored to have you with us today, sir. A very welcome to you. Today, he will be speaking on a fascinating and futuristic topic, nature's smart nanocarriers for next generation nanomedicine. This topic explored how nature itself can inspire and provide intelligent solution from drug delivery. Nanocarriers, which operate at a microscopic level, are revolutionizing how medicines are delivered in the body, making treatment more targeted, efficient, and with fewer side effects. By combining natural system with nanotechnology, researchers are opening new frontiers in medicine, paving the way for more precise and effective therapies in the future. It's a glimpse into the next generation of healthcare innovation. So without any further delay, please join me in giving a warm and enthusiastic round of applause as I invite Dr. Abul Kalam Azad to the stage. The stage is all yours, sir. Please, sir, unmute yourself first.
Uh, Thank you so much. Am I audible?
Yes, sir, you are audible.
Can you see my presentation slide?
Yes, sir.
Okay. Thank you so much and good afternoon to everyone. And thanks for Prof. Kofil to invite me to present the share the idea and my experience about the nanotechnology era. So I would like to share the NASA's smart nano carriers for next generation nanomedicine purposes. So what is the NASA's smart nano carriers? Actually, this NASA smart nano carriers is available around us, and the synthesis and the availability of the raw materials is also very cheap and uh, cheaper than the synthesis on. So this NASA smart nanocarriers usually we can drive from biological source or bio-inspired sources. So it can possess intrinsic targeting or responsive, and it can be biocompatible and self-assembly properties, and that properties make them attractive for next generation nanomedicine. For example, exosome and extracellular vesicles is one of them. So this is a self-drive vesicles used for intracellular communication to deliver biologicals to therapeutic purposes. So naturally, it carry the proteins, lipids, RNA, and maybe synthetic drugs can encapsulate using the exosomes or extracellular vesicles. Liposomes is one of them which can be bioinspired phospholipid vesicles, and it is mimic to the biological membranes for the any product which can be delivered tropically and easily can be encapsulated using the hydrophilic or hydrophobic drugs. So this is the main advantage of liposomes that can be encapsulated for both types of drugs. This is another carriers that cubosomes naturally found from plant leaf sources or any other plant sources, and this is the attractive nano carriers to deliver the synthetic medicines. Hexosomes is one of them that naturally we can find, and it has inverse hexagonal liquid crystalline nanoparticles structure that is have a unique characteristics of the hexosomes. So it's useful for controlled drug release and membrane fusions activity. Cell membrane coated nanoparticles that we can coat the membrane of the cell, for example, the red blood cells or platelets or cancer cells or immunocells to use for the anti-cancer drug delivery or immuno evasions or biometric targeting properties purposes. You can see another one called virus-like particles. These virus-like particles actually used for the vaccine development or any non-infectious viral capsid structures. It can use for the any vaccine delivery, and it has highly uniform and efficient for gene or any synthetic drug delivery as well. Protein-based nanocarriers. Protein-based nanocarriers also one of the NASA's-based nanocarriers, for example, albumin, ferritin, gelatin, or silk fibroin, and it has a.
Sir, sorry to interrupt you. Sir, currently your screen is not visible. Kindly reshare it.
Oh, okay, thank you.
Yes, sir, now it is visible.
Okay, can see now? Can you see the slide now?
Yes, sir. Yes, sir. It is visible
Okay,
now.
okay, thank you. So the next is polysaccharide based nanocarriers. I believe that most of the scientists, those who are working for drug delivery systems development, they are using a huge amount of polysaccharides. For example, the chitosan, alginate, dextran, or hyaluronic acid, and it has a unique properties that is biodegradable and mucoadhesive. So anything that we can deliver in the large intestine, or if you want to develop a drug that is acid sensitive, can be used for those polymers. Lipoprotein based nanocarriers, for example, the HDL or LDL inspired particles. Actually, we have used a lot for the drug delivery purposes as a nanocarriers, and it can be found naturally from the human blood or animal blood, and it has a useful for cardiovascular and cancer targeting drug delivery. Next is bacterial outer membrane vesicles. This can be outsourced from the naturally secreted by bacteria. For example, E. coli, we can easily fermented and collected the vesicles, and it has an emerging source for vaccine development and immunotherapy. DNA, RNA nanostructures. We have used for vaccine delivery for DNA and RNA, and also for like neurodegenerative disease purposes. So bioengineered nucleic acid assemblies and programmable and precise and responsive delivery platforms we are using for DNA and RNA. Peptide-based nanocarriers is one of the NASA nanocarriers. It has self-assembling peptides forming nanofibers, nanotubes, or vesicles form. So functional for drug, gene, or vaccine delivery. Plant-derived nanovesicles, also one of the NASA-based nanocarriers, isolated from fruits, vegetables, and medicinal plants. And it has a unique properties that have no toxicity and is easy to scalable in a production potential. Microbial fungal nanocarriers. So biofabricated nanoparticles or vesicles from microorganisms and sustainable and eco-friendly synthesis routes. Biomineralized nanoparticles is also naturally found and in an inorganic and organic form. So this magnetosome is one of the biomineralized nanoparticles, and it has a useful in imaging, for example, MRI or any other imaging technology, and targeting and nanotheranostics for cancer treatment. So this actually magnetosome can be used for a theranostic purposes. So it can deliver the anti-cancer drugs and at the same time can detect the cancer cells for cancer detection. In overall, we can make a practical umbrella to group all the NASA-based nanocarriers into 6 groups. One is cell-derived carriers, lipid-based carriers, bio-macromolecule-based carriers, genetic material-based carriers, microbial plant-derived carriers, and biohybrid or inorganic natural systems carrier. So I just want to share some of the existing treatment purposes that use for exosomes, cell-drive carriers. siRNA delivery to the any CNS disorders or brain targeted drug delivery purposes we are using, and cell membrane coated nanoparticles, for example, red blood cells membrane coated PLGL nanoparticles, very commonly using. Bacterial outer membrane vesicles, Neisseria meningitis is one of the vesicles that used for vaccine delivery. So if you observe this graph, you can see how the engineering exosomes are target therapy tools, using as a target therapy tools for precision drug delivery. So it has a unique property that is easily adaptable by our body and have no toxicity and can extend a drug half-life. So it will give a longer therapeutic effect. Lipid-based carriers like liposomes, cubosomes, and hexosomes are used currently. Doxil as a drug that is used for liposomes as a coated, and cubosomes, monoline-based cubosomes for anti-cancer drug delivery are currently using, and phlanthorel hexosomes for sustained release therapeutics are using for currently. So lipid-based carriers are using the structure like both you can use for hydrophilic and hydrophobic in nature drugs. Both drugs can be used for liposomes or cubosomes and hexosomes. Biomacromolecule based carriers, for example, the protein nanocarriers. So Abraxane is one of the drugs that used for delivery purposes coated by the protein nanocarriers, and polysaccharide nanocarriers like chitosan nanoparticles for mucosal drug delivery because it has a mucoadhesive properties. Peptide nanocarriers also using for its unique properties as a self-assembling peptide hydrogels. So you can see the albumin, protein, and drug payload. So you can load the drug into the albumin, and it can deliver to the Abraxane, and it already FDA approved anti-cancer drug. Genetic material based carriers, for example, DNA, RNA. So DNA origami drug carriers very commonly using, and RNA, siRNA loaded RNA scaffold also using for precision medicine application. So you can see the DNA scaffold, and you can load the drug into the DNA, and it can be easily delivered to the targeted cells for cancer therapy. Plant microbial drive systems, for example, a good example, ginger drive extracellular vesicles are using for the exosomes as a nanovesicles, and fungal nanoparticles, for example, from yeast drive vesicles are commonly using, and it has a high value for scalable production opportunities. So if you see here, the ginger we commonly use in our cooking purposes every day. So it has a extracellular vesicles for anti-inflammatory and anti-cancer therapy. So biohybrid and inorganic natural systems, for example, the magnetosomes for MRI guided therapy. So we are also very much using for MRI guided therapy for cancer purposes as a nanotheranostics for the diagnosis and treatment at the same time. So biohybrid, you can see the lipid membrane and magnetic iron oxide and membrane proteins, which give the extra advantage to detect the cancer cell and to deliver the anti-cancer drugs. So the key take-home message for you, the NASA-inspired nanocarriers are revolutionizing the pharmacy by addressing drug development and safety concern and global health challenges, and is a better option instead of synthesis nanocarriers, synthesizing environmental issue, the chemical used, and the pricing also high. So to synthesis from the natural sources will give you the pricing cheaper than the synthesis on, and low toxic, easy to decompose in our body. So I think this time for us to focus on the nasal nanocarriers based drug delivery for anti-cancer and neurodegenerative diseases. Thank you so much for giving me an opportunity to share the recent development and opportunities for nano drug development. Thank you.
Thank you so much, sir. That was wonderful and insightful presentation. I am sure everyone has gained valuable knowledge from this session. Now, let us move ahead to our next speaker. Ladies and gentlemen, the energy in this conference continues to grow stronger with every session, and we are truly fortunate to be part of such a rich academic exchange of ideas from across the globe. As we move ahead, we now enter into a highly modern and forward looking area of research that connects biomedical science with cutting edge technology. It's my great pleasure to introduce our next distinguished speaker, Mr. Alexander Idoku from the Department of Biochemistry, Faculty of Basic Medical Sciences, Federal University of Health Sciences, Nigeria. Mr. Idoku is a promising researcher and academician in the field of biochemistry with a strong focus on innovative approaches in therapeutic discovery. His academic interests reflect a deep commitment to addressing global health challenges through scientific innovation and interdisciplinary research. His work particularly highlights the integration of emerging technologies into biomedical sciences, making his contributions highly relevant to today's evolving research landscape. We are truly delighted to have you with us today, sir. A very warm welcome to you. Today, he will be speaking on a very contemporary and impactful topic, role of AI in anti-obesity drug discovering and repurposing, evolving opportunities for precision therapeutics. In today's era, artificial intelligence is transforming healthcare and drug discovery at an unprecedented pace. From analyzing complex biological data to identify potential drug candidates, AI is making the process more faster, more accurate, and more cost effective. In the context of obesity, a major global health concern, AI is opening new pathways for discovering and repurposing drugs, enabling more personalized and precise therapeutic approaches. So, with this session, we will provide us with valuable insights and how technology and medicine are coming together to shape the future of precision healthcare. So, without any further delay, please join me in giving a warm and enthusiastic round of applause as I invite Mr. Iduko to take the stage. The stage is all yours, sir. Please.
Sir, please turn your camera and mic on.
Can you hear me now? Sorry, am I audible now? Can you hear me?
Yes, sir, now you are audible.
Okay, sorry for that mix up. Yeah, is my screen visible? Can you see my screen?
Yes, sir, it is also visible.
Okay, thank you. I want to thank the organizer for the opportunity to do this speech or present on artificial intelligence in anti-obesity drug discovery and repurposing, evolving opportunities for precision therapeutics. Here are my presentation outlines. Basically, because of the global obesity burden and pathophysiology, there is an urgent need for novel therapeutics that are channeled towards obesity management. And this is because obesity has been reported to have gotten up to a very alarming, pandemic proportion of about 650 million adults classified as obese by the WHO. Now, talking about the health implications, obesity increases the risk of type 2 diabetes and cardiovascular diseases and certain cancer. And we all know that type 2 diabetes is another challenge, a huge challenge that combats people's health today. And so managing obesity could also mean a long way of eradicating or preventing type 2 diabetics. This obesity challenge has persisted basically because of failure in the present pharmacotherapies, particularly Olistat and some other chemotherapies that have been in the market that are glucagon-like peptide receptor agonists, oriented. And a lot of complaints have been about them. For instance, limited efficacy and undesirable side effect. Besides, obesity also has a great impact economically, globally, of about $2 trillion annually. Now, where does a biochemist come in? Now, where does a scientist come in? Looking at the challenge of obesity, obesity biochemically is actually a complex metabolic dysregulation involving the adipogenesis, that is the production, excess production of lipids in the adipose tissue, and the lipolysis and appetite signaling pathways. And all this actually require attention in the right, approach. Despite this, over the years, the traditional approach has experienced a very slow, advance, and more so, the process of getting these therapeutics are quite expensive. Now, beside that, we go further, we look at the pathophysiology of obesity. Obesity is described as excess fat accumulation in the adipose tissue by the adipocyte. And this very issue is setting because of high appetite and food consumption. And it's also resulting from the underutilized calories that have been accumulated. And, the individual keep on eating, and the calories are being accumulated in the adipose tissue, and that will result in enlargement of the adipocytes. Now, when the adipocytes become enlarged, the next thing that will happen, it will result in chronic low-grade inflammation. This is so because the pro-inflammatory adipokines are released into circulation, and it will result in hyperlipidemia, and the condition becomes, an oxidative stress condition. And in this case, the pancreas will become affected, where insulin will be resistive, and then it will result in decreased feeling for, food. I mean, satiety, and the individual keep on eating and all that, and the process continue to go on for the accumulation of more fat. Now, there are receptors that should be targeted for the development of anti-obesity drugs. These are, but not completely exclusive. There are other ones. We can also have some, but I selected these key ones. They are the glucagon-like peptide 1 receptor, the glucose-dependent insulinotropic polypeptide receptor, the peroxisome proliferator activated receptor, the melanocortin 4 receptor, and the uncoupling proteins that are involved in thermogenesis and energy expenditure. A good example is the BAM15. Beside that, there are enzymes that should be targeted that are directly involved in one, metabolic site or the other that should be involved, targeted for this anti-obesity drug, development. They include the pancreatic alpha amylase and intestinal alpha glucosidase. These enzymes are key enzymes that are associated with carbohydrate, particularly starch metabolism, where the starch is broken down to oligosaccharide by alpha amylase, and then alpha glucosidase breaks down the oligosaccharide to simple sugar like glucose for better homeostasis of sugar. Then beside, there are these other groups of the phosphodiesterase that also recently are emerging for obesity treatment, because they are connected with energy homeostasis, thermogenesis, and so they are also good target for anti-obesity drugs. Then we also have the pancreatic lipase and the hormone-sensitive lipase, and they are also associated with the regulation of fatty acid in the body, in the adipose tissue and in the liver. They help to break down triacylglycerol, and when energy is needed as alternative source from the adipose tissue. So they are also good target for obesity drug development. Then we also have fat mass and obesity associated protein. This is a protein that has its association also with fat accumulation, and targeting this very protein by inhibiting it, looking for good inhibitors, can also pave way for obesity drug development. Then we also have the protein tyrosine phosphatase 1B. These are enzymes that recently have been researched on. They are known for their negative regulative feedback mechanism on leptin and insulin. And so when these enzymes are activated, it tends to make leptin to become insensitive to cell. And so targeting this very enzyme could also go a long way to improve glucose metabolism and absorption, and then also energy need for the body. Then we also have the HMG-CoA reductase. It is known to be the rate limiting enzyme in the biosynthesis of cholesterol. So targeting it and inhibiting it can also result in the stoppage of the production of mevalonate, which precedes to cholesterol production. So excess cholesterol can be inhibited by inhibiting this enzyme. So it's a good target. The last one is acetyl-CoA carboxylase. Is another good enzyme that also finds its place in glycolysis, in production of glucose. So these are targets that could be targeted in looking at how anti-obesity drugs can be developed. Now, talking about drug discovery and development is simply identifying, designing, and developing new therapeutics that are drug-like to manage diseases. It cuts across several critical stages, including target identification, compound screening, led optimization, preclinical studies, and clinical evaluation that emerges as an optimal target should be effective, should be safe, and should be clinically viable. That is to say, it must be a druggable candidate. There are several methods available in discovering new drugs. However, the most significant that have been, used so far, tested to be more significant are the structure-based drug design and ligand-based drug design approaches. Now, talking about traditional drug discovery, what are the challenges? It is commonly known that before the onset of this AI-aided drug discovery, traditional drug discovery goes through a laborious process taken through experiment, and most times they fail. That it has high attrition rate. That is approximately 90% of candidates that are druggable fail during clinical trials. Beside that, it takes a long time and it is quite very expensive. And the challenge of complexity of metabolic system of diseases also is a challenge. And beside that, the side effects of some drugs also is a challenge in the traditional drug discovering, process. Now, beside, we also know that since the emergence or since the emergence of AI drug discovering process, a lot of progress have been encountered. Looking at this graph, we can see here that success rates between traditional and the AI assisted is far different. You can see 30 for the AI and 10 for that of the traditional. Talking about cost in millions of dollars, $2,000 million can go for a particular drug discovering using a traditional, but it will only take just 500, for that of the AI. Then talking about time in years, it can take 12 years compared to that of 5 years. So AI drug discovering is actually quite, it has a high speed and it has a high precision, attachment to it. Now, the AI revolution in pharmacology industry, what do we see? The AI actually is simply the use of algorithm, that is deep machine learning, to perform tasks that typically require human intelligence. Its capabilities can be seen because it has a pattern of recognizing high dimensional data. It can predict molecular behavior of compounds and of target. It can also simulate biological interactions where you have cell and cell interacting together, and that gives a good understanding of how the disease can be tackled. Besides, its relevance could be seen where it is modeled, where protein and ligand, you model the protein and ligand interactions and then predict the binding affinities and simulate metabolic pathway. And this makes it faster than the wet experiment or what we call the traditional method. Besides that, it has different stages in its development or process of drug discovery. We have the first stage is a target identification. At this point, you have to predict the protein and ligand interaction using AlphaFold and DeepChem, software. Then you go further to hit identification. When this first stage has gone, you can now look for the hits, the compound, in this case, the small molecule that is acting as a drug candidate. It emerges as the hit. You do that through virtual screening. Then that leads to lead optimization, obtain the compound to get the best of its property. And then you now proceed to preclinical testing where the admin test prediction is being taken, and then finally to clinical trials. And the speed, like I said, is faster than the other traditional method. So because of time, I'll quickly go to selected key molecular targets and how AI can be leveraged for anti-obesity therapeutics. There are several of these key molecular targets, but for this, I'll quickly run through just two of them because of the work we have done on those two molecular targets. The first is the glucagon-like peptide 1 receptor. The role it plays, as it has to do with obesity, is satiety and insulin secretion. Then how does AI comes in? AI will enable the design of oral small molecule instead of the peptides. Peptides are bigger molecules. And this has been achieved on the drug semaglutide. It's already being used or probably has passed its clinical trial. Then the second one I shall be talking on is the fat mass and obesity associated protein. It is known to be associated with fat metabolism and glucose homeostasis. And the role is to design selective inhibitors to this protein that has the ability to reduce weight gain and increase thermogenesis. So this is just one of the work we have done on glucagon-like peptide 1 receptor. We were able to predict the binding mechanism, molecular binding mechanism, using honey and lime juice flavonoids as our adjunctive remedies against obesity and diabetes. And in this study, we discovered that using machine learning, AI, we discovered that the protein, this very receptor, has two binding sites, port A and B, where these small molecules, the flavonoids, were able to attach themselves and bind to. And actually, this gives a great insight because it was possible because of the amino acid residue that were in these various active sites of the protein. And we were able to come up to know that these molecules, they are agonists on this very protein or receptor, the GLP-1 receptor. Like for instance, in the liver, they can reduce gluconeogenesis and steatosis. They can also reduce gastric emptying and gastrointestinal motility in the intestine. And in the brain, they have the ability, acting as agonists on this receptor, to reduce food intake, reward behavior, and reduce palatability. And in the muscle, they can improve insulin sensitivity and glucose uptake. And while in the pancreas, they can also increase insulin secretion, insulin synthesis, and decrease glucagon secretion and apoptosis, and also improve beta cell survival. And the other one, like I said, is the fat mass associated protein, the FTO protein. We're also able to target this protein looking at the same flavonoids biomolecules. They were able to act as inhibitors to this protein, to inhibit this protein, and therefore exert their anti-obesity ability. And this was possible because of the hydrogen bond formation around, contributed by this the amino acid residue in the protein. Now, what is then drug repurposing? Drug repurposing is identifying new therapeutic indications for existing approved or investigational drugs. It's a new area in AI, aided drug discovery, where existing drugs can be repurposed for better pharmacological, purpose. Now, these are some of the examples that have been, done. For example, we have drug repurposing of metformin. AI analysis of patient data confirmed and defined new mechanism for metformins to have weight loss effect beyond glycemic control. So it has been repurposed for the purpose of treating obesity. Now, we talk about the value proposition. Now, what AI is able to do is to accelerate, is to result in cost effectiveness. And then it's quite innovative because it uncovers non-intuitive drug targets that have not been known before. And so basically to help in drug discovering. And then it enhances success rates because there are better prediction of toxicity and all that. And then also data utilization, because it leverages on big data from omics, omics technology that includes the genomics, the proteomics, and the metabolomics. Now, what are the ethical concerns and limitations of the use of AI? The first that comes to mind is this problem of garbage in, garbage out, because AI relies on high quality standard biochemical data, which are often fragmented. So it may not get everything and then put everything together. So that is a problem. Beside that, there is this black yeah, it becomes very difficult on AI. It becomes very difficult to interpret how AI model reaches a specific conclusion. And beside that, experimental validation also is a challenge because AI prediction must still be validated in wet labs using in vivo and in vitro study. And it is said to be biased. And then intellectual property challenge is another question to said who owns the right to AI generated molecule. In summary and in conclusion, obesity is a complex biochemical disorder requiring advanced solution. Using AI, we can confidently look at a promising approach in transforming the problem of anti-obesity drugs by expediting the drug discovery process and enhancing
I hope everyone found that session as interesting, as informative as I did. So, moving forward, it's my great honor to introduce our next distinguished speaker, Dr. Suraya Abdul Rahman, Dean of the Faculty of Pharmacy at MAIWP International University College, Kuala Lumpur, Malaysia. Dr. Rahman is an accomplished academic leader, a respected visionary in the field of pharmaceutical science. As Dean, she has played a pivotal role in advancing academic excellence, research, and professional development within the pharmaceutical community. Her expertise and leadership in areas that intersect science global standard have earned a great record and respect. We are truly privileged to have a personality with us today. Ma'am, a very warm welcome to you. Rahman will be speaking on a very important and topic, halal pharmaceutical critical in today's global healthcare. The concept of halal pharmaceutical increasing importance. It's not only addresses religious and ethical considerations, but also emphasis quality, safety, and transparency in the sourcing and production of medicine products. Understanding the critical ingredients involved along with their compliance to a halal standard is in ensuring inclusivity and trust in healthcare system worldwide. This session will surely provide us with valuable insight into this crucial and evolving. Without any further delay, please join me in a warm and enthusiastic round of applause. I invite Dr. Suraya Abdul Rahman to take the stage. The stage is yours, ma'am. Please, ma'am.
Thank you, Ms. Chairperson. All right, good day everyone and good afternoon. Today I will be presenting on halal critical ingredients in cosmetic and pharmaceuticals based on a qualitative document analysis focusing on ingredients with doubtful or uncertain halal status. Let me share my slide first. Can you see my slide? Can you see my slide?
Yes, Prof.
Okay, thank you. Slideshow slow. Okay. Okay, so good afternoon, everyone. Today I will be presenting on halal critical ingredient in cosmetic and pharmaceutical based on a qualitative document analysis focusing on ingredients with doubtful and uncertain halal status.
Sorry to interrupt you, ma'am. Please
Yes?
share your screen first. Sorry to
Oh.
interrupt you. Please share your screen.
I already shared.
Now it's visible.
It's not visible? Huh?
Now it's visible. You can continue.
Okay. All right. So this topic is highly relevant because modern cosmetic and pharmaceutical products often contains hidden ingredients derived from complex supply chain, making halal verification increasingly challenging. Okay. Do you see the next slide? See the next slide?
Yes.
Okay. All right. So as a background information, I provide some definition here. What is halal? Halal means permissible. It is a clear guideline from the Islamic Sharia law according to Quran and Sunnah. Halal means any ingredient which is permissible because it comply with the Quran and Sunnah requirement. And in opposite is haram. It means impermissible, which strictly prohibited, for example, porcine, carrion, or canine, which and their derivative, which is strictly prohibited due to Quran and Sunnah Sharia law. And in between, there's a shubha or doubtful ingredients. The majority of halal critical ingredients falls under this category because the source of the ingredient usually unknown or doubtful because some ingredient used in cosmetic and pharmaceutical, they may come from animals as well as from plant. So they falls under critical ingredient because their status is doubtful. Okay, so the next slide shows the main ingredient which is prohibited under the Sharia law. First category are porcine and non-halal animals, for example, pigs or carnivorous animals, birds of prey. So according to Sharia law, they have categorized what other animals are prohibited or considered haram. And improperly slaughtered animals, even though they are allowed to be eaten like chicken and all that, but it is still considered haram if they are not slaughtered properly according to the Sharia Muslim law. Human and blood substances are also haram. And intoxicant like hama or alcoholic drinks in products containing liquor or intoxicating alcohol also considered haram materials. So by definition of halal pharmaceuticals, what is halal pharmaceuticals? In Malaysia, they have launched the Malaysian standard MS2424, which define the requirements or lay out the requirements if you want to apply for halal certification for halal pharmaceuticals. So under this nation standard MS2424, halal pharmaceuticals is defined as pharmaceutical product which do not contain any impurities or najis, we call it in Arabic. And also they do not contain any unlawful animals, do not contain human derivatives, and their processing, production, storage, and all that are segregated between non-halal and halal products, and the equipment used to prepare the products are using equipment not contaminated with najd. So for product to be halal, the whole supply chain of the production of pharmaceutical products from the origin until delivery to consumer must be clean and not contaminated. So at every transfer point in this production or halal value chain, a material must be verified that they do not contain any non-halal ingredient, and a failure at any single points of this chain will nullify the entire downstream process, will cause the product to be non-compliance to halal requirement. So this next slide is just to highlight that on top of the raw materials, the production process, the packaging materials also may pose a hidden non-halal risk to the finished product if you want to apply for halal certification. For example, the production of bottles, even though it's chemical polyethylene bottles and polypropylene bottles, they may contain common lubricants or slip agents that, for example, they use calcium, aluminum stearate, salts to provide lubricity when they want to produce the bottles. These stearates may actually come from animal-based ingredients. So the halal violation here is the stearates are frequently derived from beef tallow, which is not slaughtered according to Sharia law. So finish with the background information. Now we zoom into the research that I would like to share today. Is about demystifying doubt, meaning we want to find out what are the common halal critical ingredients used in cosmetic and pharmaceutical production. So the objective of the research is to identify and map critical ingredients commonly used in cosmetics and pharmaceuticals. And the research analyze, or firstly, the data is collected from reviewing a total of 238 documents or articles. And upon filtering the quality and the credibility of the documents, only 55 documents in the end we used to conduct or to analyze for our results. And from the analysis, the finding that we found from the 55 documents that we analyzed, we found 67 critical ingredients identified, and these carry hidden sourcing or jurisprudence risk. So as per the methodology, how the research is conducted, it is a document review, qualitative document review. Information are sourced from Scopus, Google's, and Gray literatures. So documents are sourced using a certain search strings, and also from looking at the relevancy of the document to our research objective. As mentioned, from the data collection, we managed to collect 238 total documents, but after quality appraisals and filtration, we only include in the final analysis 55 reports. So from the finding of the results, there are two critical ingredients can be divided into two reasons. Okay, the raw ingredients or the ingredient is considered critical because of two main reasons. The first one is because they are shubha, or they are doubtful in terms of their origin, meaning they are at risk of non-halal sourcing. So ingredients that are sourced from animals bovine, porcine, and lack defined information on the origin or the slaughter status of the animals. The key reason is it's considered critical. And also materials can be considered shubha or doubtful because there are no established decision or fatwa between the Islamic scholars regarding the permissibility of the ingredient. For example, ingredient based from animal urine. Okay, in the next slide, I will share with you what are those ingredients commonly used, which is based on this issue. So to resolve the issue of non-clear source, we need to verify the supply chain of the raw materials, or we can also conduct chemical testing. And for the issue of no decision or no fatwa on certain ingredients, the religious consensus must be formed among the Islamic scholars to give a decision on the status of the ingredient. So the next slide is I want to share is the distribution of the critical ingredient or the doubtful ingredient that we found from the 64 ingredient. We break down into their categories. Lipid-based ingredient represent the highest risk at 42.7%. This followed by protein, hormones, and enzymes, and other categories. Okay. So ingredient which are lipid-based drive the majority of risk because primarily lipid-based ingredient are usually sourced from animal fats. Proteins and hormones also represent high risk biological because they usually directly extracted from animal tissues. And hormones as well, although they are small in volume, they are critical ingredient, even though they use in small volume during manufacturing, like in vaccine production, they use trypsin and so on. Even though small volume, it still be can cause the final product non-halal if the source is non-halal or from porcine organs. So from the study, we analyze what are the top 10 critical ingredients that commonly used or commonly found in cosmetic and pharmaceutical products. So the first and the most common critical ingredients or shubha ingredients used in cosmetic and pharmaceutical products, we found glycerin or glycerol. This ingredient is commonly used in cosmetic mostly, but also used in pharmaceutical. We will see in the next slide what are the common function or use of this ingredient. The second one is stearic acid. It is also lipid-based ingredients. Thirdly is magnesium stearate. This is also a common critical ingredient used in cosmetic and pharmaceuticals, followed by sorbitan ester, also lipid-based, collagen, protein-based, ethanol is alcohol-based, propylene glycol, lipid-based, gelatin, animal-based, placenta, and allantoin. So these are the top 10 critical ingredients that we found from our analysis. So looking, zoom in into the respective function of this common critical ingredient. So glycerin, glycerol commonly used as moisturizer, as well in soap production, cleaning agent. Glycerin and glycerol, if you look at your cosmetic labels, it is commonly used in your cosmetic products. In pharma, it is also used in syrups and lozenges. And the risk, non-halal risk here is it may be derived from porcine fats or animal fats, which is not slaughtered according to halal Sharia law. Stearic acid is commonly used as emollient and lubricant in cosmetic, and in pharma they are used as tablet lubricant. This is often derived from animal tallow. Alpha hydroxy acid commonly used as skincare exfoliant, and in pharma as dry skin treatment. This may carry traces of haram animal fats. Magnesium stearates commonly used as emulsifier in cosmetic, and in pharma it is commonly used as tablet binder. The hidden risk is also they are potentially manufactured or sourced from non-halal animals. Alternative to that, actually, you can find a halal magnesium stearate from vegetable oil or palm oil. The next category, protein and hormones. When we look further on their function, collagen and gelatin are commonly used as anti-aging and gelling agent or capsule shell for pharma. So they are very highly critical because mostly gelatin are sourced from porcine skin or bone or from non-halal bovine sources. Hydrolyzed keratin, usually used in shampoo, usually used or sourced from feathers or porcine bristles. And in hormones, example, insulin used as diabetes treatment. Historically, it is derived from porcine pancreas. So this is an clear-cut non-halal and cannot be used for Muslim unless in life-threatening situation. Estrogen and progesterone, anti-aging, used for anti-aging or hormone therapy, also may be extracted from porcine ovaries. So basically, any biological based products directly extracted from animal tissues represents an extreme risk for halal status of the ingredient. And the next category, looking at the enzyme and what are examples of others here. Okay, so enzymes, for example, lipase and pancreatin, usually used in skin cleansing and malabsorption treatment. The risk is enzymes are frequently extracted directly from pancreas of porcine or non-halal slaughtered animals. Under the category of others of critical ingredients, example are alcohol, ethanol, and so on. So solvents such as ethanol, if considered haram, if they are taken from a byproduct of wine or production. So if the alcohol is a synthetic alcohol or is an industrial alcohol which is manufactured solely for medicinal or industrial purpose, that is halal and allowable. But ethanol, which taken from a byproduct from the production of wine or alcoholic drink, that is considered haram. Another haram or critical materials falls under the category of others is activated carbon. Activated carbon usually used in filter. If it is from bone, animal bone, and so on, is non-permissible. If the activated carbon comes from wood or coconut shell, it is considered halal lah. Okay, but activated carbon is a critical ingredient. Anyone want to use activated carbon-based ingredient need to really find the source of the activated carbon used in the product. Then the next category of critical ingredient is because there's no decision by there's no consensus among the Islamic scholars. These are the examples of the product which falls under this category. For example, is allantoin. Allantoin is a soothing skincare or disease treatment ingredient. The conflict is allantoin can be sourced from animal urine. Under certain school of thought, it allow from material or ingredient taken from urine is allowable, but under the Shafi'i school of thoughts, which is for the Muslim established scholar, they consider it as haram. Urea also can be sourced from urine, so it also same case in certain Islamic scholar is considered halal. In Shafi'i mazhab or school of thoughts, it is considered haram. Okay. So the next slides, I just want to show how do you know what are the source, how do you want to check what are the source of the ingredient. You can actually, for academic exercise, you can refer to established pharmaceutical reference, for example, pharmacopoeia. British pharmacopoeia, European pharmacopoeia, most of them have the information on the source of the materials. This study done in 2012, they look at the monograph or the information of around 3,200 ingredient which is listed in British Pharmacopoeia, and out of the 3,200 ingredient, they found that only 129 ingredients actually containing haram substance, because in the pharmacopoeia they will say what is the source of the material. So out of the 3,200, only 129 ingredient which clear cut it is from porcine, so it is a clear cut haram. But majority of them are considered masbuh or halal. So the key insight from here, a product can be perfectly passed BP monograph in terms of quality, purity, and efficacy, but it is still can be haram lah. So this slide, in order for a pharmaceutical company to apply for halal certification in Malaysia, because Malaysia they allow to apply halal certification for pharmaceutical products, the company need to provide a clear traceability on the source of the raw materials. So how do you prove that the source of the ingredient is halal? You need to provide a complete documentation. For example, provide a halal certificates of the raw ingredients that you want to use in the production of pharmaceuticals. And the halal certificates must be issued from recognized global Islamic bodies. And on top of that, you need to also declare your compliance to the halal certification requirement. You need to provide the specification of the product, the process flow, certificate of analysis, material safety data sheet, a lot of documents you need to provide on top if you do not have halal certificates to prove that your raw ingredients is halal, and also maybe manufacturing process flow and records. So another alternative, which also required for certain critical ingredients like gelatin and collagen in Malaysia, our JAKIM, which is the authority who issue the halal certification, require lab testing to establish that there's no porcine DNA in the raw materials. So you have a shubha ingredient, you need to do lab tests, including chromatography, PCR, electrophoresis, electrophoretic, spectroscopic, and so on, do chemical analysis, and verify that there's no non-halal markers detected on the raw materials. But on top of the halal test, the finish or lab testing, also to produce halal certified pharmaceuticals, the whole production of the whole process of the production of the pharmaceutical product need to be clean and pure, and there's no contamination. So from pre-production to production and post-production must already integrate the element of halal requirement, mean they need to control from raw materials until finished product. So we call it a halal built-in system or halal assurance system. So all the process from beginning to the end must be clean or do not contain any non-halal contamination. So the key takeaways from this sharing, okay. So from the research, we identified 64 critical ingredients that commonly used in cosmetic and pharmaceuticals, because they rely heavily on ingredients with unclear origins, which requires strict evaluation. And 95.3% majority of non-halal compliance are due to animal-based origin or derivatives, specifically in lipids and protein-based ingredient. Hence, in order to go for halal certification, the formulators must really check supply chain transparency and combine with advanced lab analysis to ensure complete compliance to Islamic jurisprudence. Okay, so I think that's all from me. Thank you for your kind attention.
Thank you so much, ma'am, for that insightful presentation. So moving forward, yes, the energy in the room is exciting, and it's wonderful to see such enthusiastic. Let us continue this momentum to get another insightful and technically here to introduce our next Yilmaz from the Izmir Institute of Technology, Materials Research, Turkey. Dr. Yilmaz, a researcher and academician known as advanced analytical and imaging techniques. Her work in the field of materials research, especially in applying micro is truly commendable.
Hi, my presentation is clear or not?
And structural bridging
Clear.
Is you just see my presentation?
Yes, it's clear.
Yes.
Is clear.
Yes,
Okay,
ma'am.
thank you very much. Thank you. I would like to thank Dr. Kapil Kumar very much for inviting me to this organization. And just I want to present electron microscopy in medicinal plants, applications of SEM and TM in pharmacognosy and phytomedicine. It's very interesting topics in nowadays. What is a macroscope? Macros means small. Scope it means look at Greek. Look at its origins is Greek. What is a microscope? There is so many little objects that human eyes can't be able to see. The microscope is a tool to see minute objects and consisting of lens or combination of lenses. Historic figures in macroscopy. I want to see these scientists. Antony van Leeuwenhoek is the first macroscopist. Then Robert Hooke, Ernst Abbe, Ernst Ruska, and Richard Feynman. The first component microscope is Zacharias Jansen's is designed the first microscope. It seems that the telescopics, I think, and the other microscopist is Galileo. Galileo developed a compound microscope, and he called is the microscope Occhiolina. It means that little eye. Is very interesting designs. The first use of term cells, Anthony van Leeuwenhoek. He writes a Micrographia. And living cells first seen and Anton van Leeuwenhoek, white blood cells, red blood cells, yeast cells, sperm cells, bacteria cells. Micrographia is very important for the biological sciences. Modern compound microscope is Ernst Abbe and Carl Zeiss invented this microscope. In 1886, and then transmission electron microscope is very developed, very modern microscope. Ernst Ruska and Max Knoll designed and built the first transmission electron microscope based on an idea of Leo Szilard. Fast contrast microscope is invented by the Zernik. Scanning electron microscope is three Americans, Dr. Zverkin, Dr. Hiller, and Dr. Snyder, described at through SEM. Confocal laser scanning microscope is Thoms and Christoph Kramer developed the practical confocal laser scanning electron microscope. This picture is view of a nucleus of bone cancer cell using normal high resolution fluorescence microscope. Scanning tunneling microscope is another name is AFM. George Binning and Henry Rohrer invented scanning tunneling microscope. This picture is we can see our eyes in 1 meter and 100 micrometer, and light microscope is we see the 100 micrometer and 100 nanometer, and electron microscope is 10 micrometer, and we see the 1 nanometer. It's very little. We can see the electron microscope is viruses, DNA and glucose and atoms. It's very little things, you know. And this is another picture is scanning tunneling electron microscope is we see the atoms 100 picometer is very little. Transmission electron microscope is we see the 100 micrometer and 100 picometer. We see the atoms by the for example fingerprints. What are SEM and TM? SEM is scanning electron microscope. TM is transmission electron microscope. We can see the scanning electron microscope is surface morphology, 3D imaging, coupled with EDX for elemental analysis, ideal for example, plants. We see the trichomes, we see the pollen powder analysis and another as. we see the imaging depth is surface. TM transmission, TM is transmission electron microscopy. Internal ultrastructure and lattice imaging is very important. Transmits electrons and through ultra-thin specimens, reveals internal organelles and nanostructures, selected area electron diffraction patterns, confirm of crystallinity for nanoparticles. HRTM results, we see the atomic lattice springs. They are very important techniques, and they are routinely combined by FTIR and XRD to provide complete structural, elemental, and chemical characterization of medicinal plant materials. It's very important for the research. Why electron microscopy is important medicinal plant research? Global reliance on herbal medicine, who estimates of percentage of age of the world population depends on plant-based medicine for primary healthcare needs is very important. Adjunctation quality of herbal drugs is very important. SEM distinguishes genuine medicinal species from adulterants and substitutes traded under the same name. And nanoparticles is very important in nowadays. Rapid growth in green nanoparticle synthesis. Plant-mediated nanoparticle synthesis is one of the fastest growing areas. TM, SEM are primary characterizations. They are very important. And bridge, connecting morphology and nanoscience. EM bridges plant morphology, phytochemistry, and nanomedicine, enabling drug delivery and bioactivity studies. Application span species adjunctation, tissue ultrastructure, nanoparticle characterization, drug delivery, pollen taxonomy, and phytochemical localization. They are very important analyses. You see. Tissue ultrastructure and morphological characterization. For example, trichome diagnostics. SEM of, for example, 5 Artemisia species, resolved glandular or non-glandular trichome morphology. Density are reliable characters for species discrimination. It's very important. Quantitative microfeatures, for example, heliotropium rarifolium, setos glandular trichomes size is very important. The distinguish of the species. Pollen and seed micromorphology. SEM, for example, of Svertia criarita revealed reticulate alveolate seed coat and radially symmetrical in perculate pollen, useful for species identification. Leaf ultrastructure, Genipa americana, SEM described cuticle trichomes and surface tissues. Morphological markers is very important, botanical identity and storage tissues arrangement. For example, SEM of plant leaf surface showing trichomes and stomas. Some imaging showing stomata and trichomes on plant leaf surface is very important. Some characterization of cuticular and trichome morphology as botanical identity markers. For example, stomatas, for example, vascular areas, trichomes, and another as you just see light microscope and electron microscope images. Some of Svertia criata showing reticulate alveolate seed coat and inopeniculate pollen. These images are seed coating and its pollen morphology. Its type of pollen is inoperculite. The morphology pollen is very important. SEM is very important to learn about the pollen morphology. Adjuvantation and herbal drug standardization, comparative light microscope and SEM studies distinguished 3 commercial confused sources sought under the same trait of Halion, demonstrating that microscopy reliable discriminate genuine medicinal sources from substances. Pharmacopoeia standardization is very important. For example, SEM integrated with FTIR XRT for complete the standardization profile. And we have some EDX elemental fingerprinting. For example, this species is Olea europea. The another name is olives. Elemental detected is calcium, silicium, magnesium, carbon, and oxygen. EDX signals complement morphological markers for drug standardization and batch uniformity checks. Calcium, magnesium crystals in plant tissue, and we just see the under microscopy is very important. Mapping and phytochemicals and cell wall architecture, secondary metabolite storage sites, TM localizes secondary metabolites in vacuoles, idioplasts, glandular trichomes, critical for understanding pharmacological activity distribution within plant tissues is very important. Crystals and inclusion mapping is important. Some studies routinely identify calcium oxalate crystals, pitted vessels, and storage parenchyma in viola and primula species, for example, indicators of the phytochemical localization. Elemental mapping with via some EDX analyzes, calcium, silicium, potassium, carbon, and oxygen quantified, for example, Thea, basil, or another plants. Elemental patterns inform mineralization roles in bioactivity and safety assessment. SEM revealed processing depends starch granules, reorganization and breakdown in cell walls. Metabolics identified 57 determinate alkaloids is very important because bioactive components confirmed. Microstructural change correlated with shifts in chemical composition and pharmacodynamics. Integrative SEM and XRD metabolics approach established processing quality relationships. And this is a mapping analysis. And you just see the elemental colors. For example, oxygen is orange, aluminum is turquoise, silicium is pink, magnesium is red, and we just see the elemental composition of Olea europea. Is a morphological markers for drug standardization is very important. Plant-mediated nanoparticle synthesis, SEM and TM characterization, mechanisms of green synthesis, plant phenolics and flavonoids act as both reducing and capping agents. The resulting nanoparticles are stabilized by biomolecular, confirmed by FDI spectroscopy and other analyses. Role of SEM and TM, core particle size measurements and lattice fringes for crystallinity confirmation. HR SEM, we just get surface of morphology, agglomeration, state, and particle distribution, visualization, UV with plasmon resonance, Cora-based TM, SEM size data. SEM and EDX analyzes, we just get the elemental composition confirms, for example, this sample, silver nanoparticles synthesized from plant extract. We just silver or gold nanoparticle is identity is very important. These analyses. Comparative nanoparticle characterization of medicinal plants, for example, Selaginella meiosaurus, Chrysanthemum parthenium, Osmium sanctum, and 5 medicinal plants mixed. They used of silver nanoparticles and they get 1990 in vitro inhibition and 60 anti-inflammatory. This is means Carrageen model is important for the drug sector. And another plant is silver nanoparticles. We just used XRD FTIR, UV visible. FTIR confirmed plant functional groups in reduction and stabilization. Osmium sanctum, they can use TM, UV visible FTIR, TM morphology, enhanced antibacterial activity, for example, Escherichia coli, quercetin mediated synthesis. And another model is B model, TM, XRD, and FTIR. Green root influences size, morphology, antimicrobial efficiency varies by source. TM and SEM characterize nanoparticles as bioactive agents. Plant-derived nanoparticles characterize these methods and serve bioactive agents and delivery platforms with measurable cytotoxicity, selectivity, and in vivo anti-inflammatory biomedical applications, for example, gold nanoparticles from dandelion, TM characterized, for example, silver nanoparticle formulations, lavender extract nanoemission, and Selaginella nanoparticles inhibition in Carrageenan edema model, another model, in vivo anti-inflammatory delivery. Pollen morphology, seed coat powder macroscopy. Pollen morphology, you just get some reveals exine architecture, reticulate, granular, echinate patterns. Svertia, for example, create a radially symmetrical in operculate pollen itself. Palynological data supports taxonomic classification is very important. Seed coat analyzes SEM, we get SEM seed surface, reticulate alveolate patterns. Seed coat features correlate with dispersal mechanisms and germination biology used in endangered species conservation and identification. Powder macroscopy, we just some of crude drug powder, for example, trichomes, vessels, crystals, and starch grains, and another as challenged and in the future directions, sample preparation complexity. For example, biological specimens require fixation, dehydration, critical point drying, and sputter coating, time consuming, and may introduce artifacts. High cost and expertise. SEM and TM instruments are very expensive to acquire and maintain, and skilled operations are required for data acquisitions and interpretation, processing artifacts, dehydration and fixation. Steps can alternative cell wall structure and organelle morphology, potentially misrepresenting in vivo states, limited in vivo applicability. Electron microscopy requires in very high vacuum conditions, incompatible with live imaging and hydrated biological tissues. Future directions, we hope, cryo-SEM and cryo-TM, hydrated native state imaging without chemical fixation, preserves through in vivo structure of plant cells or nanoparticles. Correlative light and electron microscopy combines fluorescence localization with electron microscopy ultrastructures and enables precise mapping fluorescently labeled phytochemical materials. AI-assisted image analysis, we think, deep learning for automated trichome counting, particle sizing, and feature extraction, enable high throughput screening and medicinal plants materials. And last one, multi-omics integration, linking electron microscope derived morphological data with metabolics, genomics, and proteomics, holistics, and understanding medicinal plant biology is very important. Integration of SEM and TM, AI image analysis is, we hope. Conclusion, we get indispensable research tools. SEM and TM are very important in modern medicinal plant research, is very important. From tissue ultrastructure to nanoscale drug delivery, characterization, wide application spectrum, applications span species documentation, quality control, nanoparticle synthesis, drug delivery, pollen taxonomy, and phytochemical localization, enhanced by complementary techniques, combination of SEM, TM with EDX, FDIR, and XRD provides complete structural, elemental, and chemical characterization profiles. Future integration with AI and cryo electron microscopy, emerging cryo electron microscopy, CLAM, and AI assisted analysis will further expand capabilities and enable high throughput medicinal plant screening. Electron microscopy continues to bridge gap between traditional pharmacognosy and modern nanomedicine in medicinal plant research. Our center images I want to show you. This is a stoma, a Laurus nobilis leaf surface. In a special mode, environmental scanning electron microscope mode, it depends on the pressures. The pressure is very high for about 100 pascals because the sample is very, sample must be very humidity. Humidity is very important because it's leaves. And another, our center SEM images, pollens and pollens. We just designed the colorful, very colorful. Our center SEM images, pollen, these we get awards of the electron microscopy congress. This is name is beans, is similar to beans. And ISTEC Center for Materials Research, we get new instruments, for example, scanning electron microscope and FIB focused ion beam systems. And we have TM, very high resolution TM, 200 kilovoltage is very high resolution. It depends of 60 picometer. And my colleagues, thank you. This is a lasers of the skin. Thank you very much for your attention. Thank
A
you.
big thank you to you, to our esteemed speaker for such a clear and impactful presentation. Moving forward, it's my absolute pleasure to introduce Professor Dr. Mahmood Arba from the Faculty of Pharmacy, Holu Olio University, Indonesia. A passionate researcher and an inspiring academician, Professor Arba has made significant contributions in the field of pharmaceutical sciences. His work, especially in advancing modern approaches to drug discovery, reflects innovation, dedication, and a deep commitment to scientific excellence. His expertise in computational studies has opened new doors in making drug developments faster, smarter, and more efficient. We are truly delighted and honored to have you with us today, Professor. A very warm welcome to you. Today, Dr. Arba will be speaking on a topic that is shaping the future of healthcare and research, the role of computational study in accelerating drug discovery process. In simple terms, computational studies are transforming the way we discover medicines. Instead of relying only on traditional methods that can take years, scientists now use advanced computer-based techniques to stimulate, analyze, and predict how potential drugs will behave. This not only speed up the entire process, but also improve accuracy, reduces cost, and brings life-saving medicine closer to reality much faster than ever before. Sound fascinating, isn't it? So without any further delay, let us dive into the innovative world of science. Please join me in extending a big round of applause as I invite Professor Dr. Mahmood Arba to take the stage. The stage is all yours, Professor, please.
Okay, thank you very much, moderator. I'm very sorry for delay for my speaking. So thanks very much for Professor Kumar who invited me for this wonderful meeting. So I am Muhammad Arba from Faculty of Pharmacy, Universitas Haloleo, Indonesia. So I think my talk today is similar to what the Dr. Alexander just talked. So drug discovery consists of so many steps, starting from target identification, and then height identification, lead identification, and so on. And this process can be done by a computational approach. Yeah. First one, I would like to present our work on identification of analog remdesivir as inhibitor of RDRP SARS-CoV-2. So RDRP is a very important drug target as it exists only in the virus and not in human. And the remdesivir is very well known for targeting this RDRP. So what we done first, we perform a molecular dynamic simulation to see the stability of the system. So we evaluate by using parameters like RAMASD, protein RMSF and ligand MRSF. And we see that the receptor backbone RNA and ligand is very stable during 1000 nanosecond. That's quite long. And then also the case is in the protein RMSF, the pattern is very stable. Here the loop are fluctuated, but the most of the interacting residue are not fluctuated. Now after that, we design a new remdesivir analog. We call it R1T, R12, and R13 by substitution H amino and methyl amino. Now and then we change the substituent here, while the rest of molecule not changed. And then we perform the molecular decaying to see how the design analog interacted with the RDRP, and we see here. This is R1T, and this is RNA, and this is R12, and this is R13. And then we see the stability of R1T, R12, and R3T. And we saw that the RMSD is very stable for 1000 nanosecond. This is a protein RMSD, this is RNA, and this is a ligand RMSD. So the analog is very stable, like the remdesivir itself. And then we perform MMGBSA calculation to predict the affinity of the analog, and we see here the R1T, R2T, and R3T has a lower affinity, lower binding energy than remdesivir. So what we have designed is so here that bind stronger than remdesivir. So here, by using a molecular dynamic simulation, we determined that remdesivir reproduce experimental structure interaction when binding to RDRP, and then we see the role of key residues such as serine 759, aspartate 790, 791 is a key residue. And then we see that the remdesivir analog bind more strongly to RDRP active site as compared to RTP based on MMPBSA binding energy. Next, I would like to show another approach, network pharmacology, what we call is network pharmacology approach for predicting anti-hepatitis C virus potential of a mangrove. So as we know that WHO estimate more than 300 million people in the world infected with the HCV, and then more than 50 million people live with chronic HCV, and then approximately 1 million new cases every year. So it's substantiated of the need to find a new cure for HCV. So the treatment, the current treatment for HCV is sofosbuvir, but the adverse effect including flu-like symptoms, blood cell abnormality, depression, and also autoimmune disorder were found at the patient. Now we proposed the mangrove unique as a unique coastal ecosystem with rich biodiversity, and then use in herbal remedies as a tradition, and many research have performed on this mangrove as a source of novel bioactive metabolite. What we do is, first of all, we analyze the mangrove Avicennia marina, and then we analysis using liquid chromatography, high resolution mass spectrometry, and then we use drug likeness screening using Lipinski rule of 5, and then we perform network pharmacology, and then we done molecular docking, molecular dynamic simulation, and in vitro test against HCV. Then result LC HRMS identified 74 compounds from Avicennia marine extract, and then from that only 4 compound violated more than one rule of Lipinski rule. So we excluded the 4 compound for further analysis. And then we perform a Swiss target prediction, and we found out about 1,500 compound related target, and more than 600 HCV related target using only chart. And then we found 88 overlapping target by using a Venn diagram. Yes. Then using some metrics like the degree, closeness, and betweenness, we've saw that the top 10 protein for this HCV, interleukin 6, STAT3, TNF, SRC, and so on. And this is the value of degree, closeness, and betweenness centrality. And this is gene ontology for top 10 cellular component, molecular function, and also biological process. And then after that, we pick the three target for the molecular docking: interleukin 6, 10F alpha, and STAT3. And we obtain that some molecule has comparable binding affinity compared to existing drug. We use a prednisolone in this case for interleukin 6. We see here prednisolone has binding affinity about -3.9, and our compound -3.4 and -7.3. And then the case similar with when docking to TNF alpha. And then we perform molecular dynamic simulation, and we see here the prednisolone more stable than our compound 63 here. And but overall the complex is stable based on the value of RMSD here. And then next we perform in vitro and anti-HCV assay, and we obtain the IC50 for the extract is about 12. 0.6 ppm. It is comparable with other study like Behbahani in 2003. The effective concentration is 10, and Beula 2012 403, and Premnatan in with effective concentration of 46. So the conclusion is this study demonstrates the potential of Marina, Avicennia marina, methylonic leaf, to inhibit HCV infection through combination network, pharmacology, molecular docking, and in vitro validation. And then we found out interleukin-6, STAT3, and TNF-alpha as core HCV-related target. And then most significant pathway is pathway in cancer, as shown in KEGG. And key compound found such as compounds 3 and compound 6,3 flavonoid, and compound 4 from alkaloid. So strong binding affinity to interleukin-6 and TNF-alpha. So this finding provide foundation for further research and develop of Avicennia marina as natural remedy for HCV. So I think this all for my presentation. Thank you for committee who has invited me, and very sorry for two times delay in this afternoon. Thank you.
Thank you so much, sir, for that insightful and enlightening presentation. What an engaging and knowledge-filled experience this conference has been so far. Each session continues to inspire us and deepen our understanding of pharmaceutical and biomedical sciences. Let us now move forward to another highly interesting and research-driven session that beautifully combines natural products with experimental science. It is my great pleasure to introduce our next distinguished speaker, Dr. Prince Ukchuwu LBO from the Department of Biochemistry, Michelle Okpara University of Agriculture, Nigeria. Dr. LBO is a dedicated researcher and academician known for his contributions in the field of biochemistry and biomedical research. His work focuses on exploring the therapeutic potential of natural compounds and understanding their effects through scientific investigation. His commitment to advancing research, especially in areas related to metabolic disorders and natural product pharmacology, is truly commendable. We are honored to have such a passionate and knowledgeable expert with us today. Sir, a very warm welcome to you. Today, Dr. LBO will be speaking on a very intriguing and impactful topic, synergistic anti-glycemic effects of turmeric and ginger methanol extracts on diabetic albino rats. The study explores how natural substances like turmeric and ginger, well known for their medicinal properties, can work together to enhance their therapeutic effects, particularly in managing blood glucose levels. By investigating their combined or synergistic effect, this research opens up new possibilities for developing more effective and natural approaches to diabetes management. It is a fascinating blend of traditional knowledge and experimental science that holds great promise for the future of healthcare. So without any further delay, please join me in giving a big and enthusiastic round of applause as I invite Dr. Prince to take the stage. The stage is all yours, sir. Please, sir. Sir, please unmute yourself first.
Hear me now?
Yes, sir, you are audible now.
Good morning. Okay. Good morning, everyone. I want to especially thank Professor Negi and Professor Kapiu Kuma for this privilege. My name is Dr. Prince Ogochukwu Alebo from the Department of Biochemistry, Molecular University of Agriculture, Umudike, Abia State, Nigeria. Today, I'll be speaking on synergistic anti-hyperglycemic effects of turmeric and ginger extracts. Okay, background. We know that diabetes mellitus is a chronic metabolic disorder characterized by hyperglycemia. It is caused by defects in insulin secretion, insulin resistance. It has been a global health burden. And there is need for
Sorry to interrupt you, sir. Please reshare your screen first.
Okay.
Presentation is not visible. Please reshare your screen again.
Okay. Trying to do that. Can you see me now?
No, sir, it's not visible. Now it's visible, sir. You can continue.
Okay. Okay. In silico study, 40 phytocompounds from Cucina longa were screened, and the molecular docking against key metabolic targets were also conducted. Several compounds showed then we have strong binding affinities that is comparable to standard anti-diabetic drugs, which suggests multi-target therapeutic potentials of this Cucina longa. In in vivo experimental design, diabetes were induced in rats, albino rats, and then we grouped the rats into normal control, diabetic untreated, metformin treated with 100 milligram per kg body weight, curcumin gingerol treated with 100 milligram, 200 milligram, and 400 milligram per kg body weight of the rats. Then the parameters that we assess were fasting blood glucose, hematological indices, histopathology of the liver, kidney, pancreas, heart, and spleen. Our key findings: there were significant dose-dependent glucose reduction, especially in 400 milligram kg body weight of the rat group. There was a reduction in blood glucose level from 229 to 50 milligram per DL, which is more important. Improved hematological parameters. There was restoration of tissue architectures and reduction in organ damage, which shows that this combination of curcuma longa, and Zingiber officinale, we are of much great importance in reduction of glucose level. So the next slide is the results. When you look at the result, you see the normal control, the 0 week and the week 2. Look at the negative control. Look at metformin, 100 milligram per kg. Then the fourth group, which is curcumin and gingerol. You see the reduction. See from the 0 week to the week 2. You see the reduction. Then you look at the curcumin, 200 milligram and gingerol, 200 milligram. You see the reduction from 268.75 plus or minus 41.79 to 54.00 plus or minus 0.58. Look at the highest dose, which is curcumin, 400 milligram and gingerol, 400 milligram. See the reduction from 229.75 plus minus 46.78 to 50.00 to 0.00. So looking at these results, you can see the efficacy of this combined or synergistic turmeric and ginger effects on reduction of blood sugar level. They showed more efficacy than the metformin, which is the standard drug. You can see it's 66, while the 100 milligram showed 56, the 200 milligram showed 54, and then the highest dose, which is 400 milligrams, showed 50. So from the results, we can see that the synergistic effects of curcumin and gingerol showed a great reduction in blood sugar level. In conclusion, combined curcumin longa and Zingiber officinale showed anti-diabetic effects, organ protective properties, hepatoprotective, renal protective, hematoprotective, and this plant shows a promising plant-based alternative therapy, which is suitable for resource-limiting settings. And we recommend further metastatic and clinical studies. Then, from our findings, we have contribution to knowledge. From these findings, it provides experimental evidence supporting the anti-diabetic potential of curcumin longa and Zingiber officinale. Also, from the findings, it demonstrates synergistic effects of curcumin and gingerol, supporting multi-target therapy over single drug approaches. Also, it shows ability to correct hematological abnormalities and protect vital organs like kidney, liver, indicating safer and therapeutic value. It also highlights potential of these plant extracts as affordable anti-diabetic agents relevant to global health challenges, especially in low and middle income countries. From our findings, it indicates comparable therapeutic effects to standard drugs such as metformin, supporting their relevance in drug development. It also strengthens the scientific basis for plant-based drug development in diabetes management. It also provides a foundation for further research into curcumin gingerol combination as safe and effective therapeutic candidates. I want to appreciate my colleagues, Associate Professor Ngozi Achi, my HOD, for providing a conducive lab and environment for this research, and also Associate Professor Samuel Onoja for assisting us in laboratory analysis. I also want to appreciate pharmacist Chidera Balihi for assisting us in silico docking, and my project students of 2025 sets. You guys are wonderful. Thank you very much. Thank you.
We are truly grateful for your valuable and thought-provoking insight. Thank you so much, sir. Ladies and gentlemen, as we now approach the conclusion of this remarkable international conference, it's time to reflect on the journey we have shared today. Filling with knowledge, innovation, and truly inspiring academic discussion from across the globe, from computational drug discovery, ethnopharmacology, nanomedicine, regulatory science, advanced microscopy to artificial intelligence in therapeutic, each session has added immense value and broadened our scientific horizons. Now, it's my great honor to invite Dr. Amit Kumar Sen, Vice Principal, Samrat Prithviraj Chauhan College of Pharmacy, Kashipur, Uttarakhand, India, to deliver the vote of thanks. Sir, we warmly welcome you to kindly address the gathering. Please, sir.
Thank you, Vasudha ma'am. Good afternoon to everyone, respected dignitaries, invitees, participants, and students. I am feeling honored to get this opportunity to conclude the webinar on revolutionizing pharmacy, addressing drug development, safety concern, and global health challenges. Certainly, this international webinar was very useful and we all got opportunity to make aware ourselves with challenges and development in the field of pharmaceutical sectors. But organizing an international conference is a challenging task. This cannot be possible without great preparation, wise working hand supports from every corner and blessings. Now, it's a time to convey our gratitude to all who have extended their supports to make this event successful. At the outside, on behalf of organizing committee, I would like to express my profound gratitude to our Honorable Managing Director and Chairman, Sir. His blessings and motivations. Sir, you are our source of motivation and energy. Honorable Managing Director of Samrat Prithviraj Chauhan College of Pharmacy, Mr. Mahesh Singh Chauhan, Sir. Chairman, Mr. Gopal Singh Chauhan, Sir. Now, I would like to thank Dr. Husne, Age University, Faculty of Pharmacy, Department of Pharmacognosy, Bornova, Izmir, Turkey, for her valuable speech. I would like to thank Dr. Alexander Doko, Department of Biochemistry, Federal University of Health Sciences, Nigeria. I also wish to thank Professor Jehra, Izmir Institute of Technology, Center of Materials Search, Izmir, Turkey, for her awesome lecture, which has been very fruitful to us. I would like to thank Dr. Muhammad Abdul Kalam Azad, Faculty of Pharmacy, University, MIWP, International, Malaysia, for his excellent lecture. I also wish to thank Dr. Prince Michael Opera, University of Agriculture, Department of International, Malaysia, for his awesome lecture. My thanks also to Dr. Suraya Abdul Rahman, Dean Faculty of Pharmacy, University, MAIWP, International, Malaysia, for her awesome lecture. I would like to thank Professor Dr. Muhammad Arwa, Faculty of Pharmacy, Halolio University, Indonesia, for his valuable lecture. I also wish to thank Associate Professor Ahmad Nazib, Faculty of Pharmacy, Universities, Muslims, Indonesia, for his knowledgeable lecture. I would like to express our heartfelt gratitude to our Honorable Director of Samrat Prithviraj Chauhan College of Pharmacy, Professor Dr. Kapil Kumar Sir, for his visionary leadership, constant encouragement, and for making this international webinar and inaugural session possible. Your guidance continues to inspire us to strive for excellence in the field of pharmacy education and research. My thanks to Ms. Vasudha Negi ma'am, Assistant Professor, Department of Pharmacognosy, Samrat Prithviraj Chauhan College of Pharmacy for her welcome speech and support. I wish to thank Mr. Manish Kumar Saxena, Assistant Professor, Department of Pharmaceutics, Samrat Prithviraj Chauhan College of Pharmacy for webinar moderate and technical support. Thank you. Thank you, everyone. Thank you so much.
At the last, I request to all the speakers to kindly turn on your camera for the collage photograph. Thank you, everyone.